Assalamu alaikum everyone, this is Abdullah Khalil and this is the platform where you can learn O-level, A-level and IGCSE biology of various UK boards. The topic that we are doing today is biological molecules from AS level biology. This is the second topic in CAIE AS biology. And as always, you should skim through the learning objectives that are given before the topics, the learning objectives provided by the board. We will start with the first topic. Now, I will share the link of these notes in the description so you can go through the details of these notes. Before we get into carbohydrates, lipids and proteins, let's first understand the concept of monomers and polymers. So what are monomers? Monomers are the single units which can join up with other monomers to make a long chain. The single unit of something. For example, the complex carbohydrates are made up of smaller units joined up together in a longer chain. Now that chain could be branched or a straight chain. This single unit that makes a complex substance by joining with other single units is called a monomer. And the complex substance that has been formed is called a polymer. So here we can see that these are some of the monomers given here, single units. But when these single units join up to make a longer chain, this longer chain is called a polymer. An example could be carbohydrate and protein. Amino acids are the single units. Joining up together, making a long chain of amino acid, that is a polypeptide. Polypeptide is a polymer. Amino acid is the monomer of polypeptide. Similarly, there are two reactions that you should be able to explain, because these two reactions are very happening in making and breaking of biological molecules. The two reactions are condensation reaction and hydrolysis. Condensation reaction is the one in which two smaller substances can join up to make a bigger substance. So in this equation, we can see that two amino acids here, this is the generic structure of amino acids. Now these two amino acids are joining up to make a dipeptide. What happens here is, as you can see OH here and H here, they can make a bond to make water. Now as water will be lost, this carbon loses one of its bond, and similarly this nitrogen loses one of its bond. So this carbon and nitrogen will make a bond together, forming a dipeptide like this. This is an example of condensation reaction. So joining of two molecules with the loss of water, that's condensation simplified. If you reverse this, it would become hydrolysis. Hydrolysis is when you break up a complex substance into two equal halves, into two substances. So when a substance is broken down by the addition of water, that is hydrolysis. Condensation, again, condensation, two molecules joining up together, loss of water, making a complex substance, that is condensation. Hydrolysis, a molecule, a complex molecule is being broken down with the addition of water. There are some examples of naturally occurring polymers given here and some examples of synthetic polymers here. For example, cellulose, proteins, DNA, these are examples of naturally occurring polymers, and we have polyethylene, polypropylene, polystyrene. These are examples of synthetic polymers. Now let's talk about the first biological molecule, carbohydrates. Carbo is for carbon, hydrates is for water. Because it contains carbon and hydrogen and oxygen in the same ratio as water, 2:1. Hydrogen and oxygen will be in the same ratio as water. If you talk about glucose, the formula for glucose is C6H12O6. So H12O6 is the same ratio as water. There are three types of carbohydrates. Monosaccharides is the monomer of carbohydrate. We have understood the concept. Monomers are the single units that can join up to form polymers. So monosaccharide is the monomer of carbohydrates. When two monosaccharides will join up, any two monosaccharides, they will form a disaccharide. Now these are all sugars. All disaccharides are sugars. Monosaccharides are sugars. And when a long chain of monosaccharide is formed, when monosaccharides join up together to make a long chain, that would form a polysaccharide. For example, cellulose or glycogen or starch. Let's look at the linear structure of glucose. This is the linear structure. As you can see here, carbon is forming the central skeleton. There are 6 carbons. The first carbon has 2 bonds with oxygen, a double bond, and the last carbon is here, H2OH. The 5th carbon and the first carbon are involved in making the ring. The first carbon will break one of its bond with oxygen The fifth carbon, where we have OH, the bond between O and H will be broken down. This carbon will make a bond with this oxygen in turn, and this oxygen will make a bond with hydrogen, forming a ring structure. Now this ring structure could be of two types. We have alpha glucose and beta glucose. If carbon number 1 has H at the top and OH at the bottom, this is alpha glucose. If H is at the top at carbon number 1, this is alpha glucose. If H is at the bottom, this is beta glucose. Now, as we have seen, this is carbon number 1. So, carbon number 1 is here. Carbon number 1. This is carbon number 2. This is carbon number 2 here. This is carbon number 3. Carbon number 3 is here. Carbon number 4. Carbon number 5. And as you can see, carbon number 6 is out of the ring. This is what is forming the ring. Carbon number 6 is out of the ring at the top for both alpha and beta glucose. This is how the numbering goes. So the ring structure is formed by a linear structure, which could be either alpha glucose or beta glucose. We can simply represent alpha glucose with this structure or this structure. But when we are forming complex substances like polysaccharides, we will just represent it with a hexagonal ring. Okay, now let's talk about the chains that are formed by glucose. There are two types of chains. Either the chain is straight without any branches. That means the carbon is making a bond with another carbon here. Two rings of glucose. Let's imagine two alpha glucose here. The carbon number 1 in the first alpha glucose is making a bond with carbon number 4 by oxygen. What happens here? This is basically hydrogen and OH. They will make a bond just like we have seen previously when we were understanding the condensation reaction. The condensation reaction is happening here. Water will be formed. And this oxygen, which now has a free electron, will make a bond with this carbon, which also has a free electron, by the way. So oxygen and carbon will make a bond, forming a disaccharide. So disaccharide is formed in this condition. A disaccharide has been formed by a bond between carbon number 1 and carbon number 4. This particular bond is called 1,4-glycosidic linkage. If a chain only has 1,4-glycosidic linkages, this is called amylose chain. This is a straight chain. But if a chain has branches like this, so what is happening here? The carbon number 6, which is present outside the ring, if this carbon makes a bond with another glucose hexagon, carbon number 1 of another glucose molecule, this particular bond is between carbon number 1 and carbon number 6. This bond is called 1,6-glycosidic linkage. So we have amylose, which is a straight chain, forming a straight chain. Only 1,4-glycosidic linkages are present. And we have amylopectin, which has 1,4 and 1,6-glycosidic linkages. Now the examples of polysaccharides, if we go back here, some of the monosaccharides are given here: glucose, fructose, galactose, ribose, deoxyribose. These are examples of monosaccharides. When two monosaccharides join, as I said, glucose and glucose, two monosaccharides, they can join up to make maltose, which is a disaccharide. Glucose and fructose can join up to make sucrose, which is a disaccharide. Glucose and galactose, both monosaccharides, can join up to make lactose. Maltose is the sugar which is formed by the breakdown of starch in our mouth by the action of amylase. So maltose is two glucose rings joined together. Sucrose is a special one. Why? Because this is the one that we use at our homes. This is the table sugar. And more importantly, this is an example of a non-reducing sugar. Rest of the sugars that we have seen here, like maltose, lactose, fructose, glucose, etc., these are reducing sugars. These can be tested with Benedict's test. But sucrose is a non-reducing sugar. You cannot test it with Benedict's reagent. Then examples of polysaccharides. When a long chain of monosaccharides is formed, it can form a cellulose or glycogen or starch. These three are the examples of polysaccharides. All three of these are actually made up of glucose rings joined together, making a long chain. Cellulose is made from beta glucose units, while glycogen and starch are made from alpha glucose units. We know that glycogen is the storage substance in animals. Glucose is stored in the form of glycogen in animals. Glucose is stored in the form of starch in plants. And cellulose is the substance that makes up the cell wall of plants. So these three are the examples of polysaccharides. Let's look at their structure and how this structure is formed. Starch and glycogen could be formed simply by repeating this bond again and again. Okay? But because it may have some branches as well, it may have 1,6 glycosidic linkages. So some of the glucose rings will be formed here as well. Amylose and amylopectin, both chains are present in starch and glycogen. Starch and glycogen are mixtures of amylose and amylopectin. So they have 1,4 glycosidic bonds and 1,6 glycosidic bonds. Okay, now let's talk about the structure of cellulose. Because it is made up of beta glucose units, and as you remember, beta glucose has hydrogen at the bottom and OH at the top at carbon number 1. Because this OH here and this OH are not in the same plane, they cannot make a bond. So in order to make this bond work, this particular molecule will have to flip. For this one, we have seen the first structure flip. The first glucose ring has been flipped. OH comes at the bottom and the bond is formed here. But again, if you can see here, OH and OH are not aligned together. Because they are not aligned together, the bond cannot be formed. So again, this molecule will have to be flipped. 180-degree flip. Actually, every alternate molecule will be flipped 180 degrees. So that the bond will be formed in this way. One bond formed at the bottom, the other at the top, then another at the bottom, then another at the top, forming the molecule of cellulose like this. Now, these are not the only bonds, these are glycosidic linkages, but these are not the only bonds present in cellulose. There are other hydrogen bonding present as well, making the cellulose one of the strongest structures in plants. The tensile strength of cellulose is compared with mild steel, so it is that strong. This is the overall structure, how the cell wall would look like. This cell wall has many cellulose fibers arranged in different zigzag patterns. If you take out one of the cellulose fibers, it looks like this, made up of many microfibrils, and each microfibril has cellulose molecules like this. Each cellulose molecule is made up of beta glucose units. These are all beta glucose units joined together by the bond that we have discussed here. So this is how the cellulose is formed. We have talked about hydrogen bonding. Let's understand what hydrogen bonding is. Basically, hydrogen bonding is the attraction of positive hydrogen with the negative end of the molecule. The prime example to understand this is water. If you look at the structure of water, this is the structure of water, okay? The bent or angular structure, the hydrogen and oxygen are forming a bent structure here. Hydrogen is partially positive and oxygen is partially negative. Why? Because hydrogen has less electronegativity, less power to attract the shared pair of electrons as compared to oxygen. Oxygen has more power to attract the shared pair of electrons. Because oxygen is negative and hydrogen is positive, there is attraction between positive hydrogen and negative oxygen all over the structure. This makes the structure very strong. A single hydrogen bond is not that strong. It's actually a weak bond. But because there are so many hydrogen bondings, the overall effect is huge. The overall impact on the structure is huge, making it really strong. Now moving on, starch is a polymer of glucose. It's a polysaccharide made up of glucose units joining to make a long chain. And that chain is actually folded to make up this coiled structure. This is the structure of starch. Starch, again, just to remind you, starch is the storage form of glucose in plants. The storage form of glucose in animals is glycogen, which has comparatively similar structure in terms of the chain, because it is also made up of alpha glucose units made up of amylose and amylopectin. But that is present in animals and this is present in plants. If you're given a solution, you have to find out if starch is present or not. Okay, you don't know if starch is present in that given solution or not. You can simply do the iodine test to find out if there is any starch. Now, obviously, iodine solution, we call it iodine solution, but it is the solution of potassium iodide, not iodine gas. When iodine solution mixes with starch, it forms a blue-black substance, a starch iodine complex. This hole present in the center is actually the right place for iodine to fit in, forming starch iodine complex, which is blue-black in color. So whenever you mix iodine with the starch, you will get blue-black color. So if there is a given sample where you have to test the presence of starch, you can simply do iodine test. You can put a few drops of iodine, which is originally brown in color, and you stir it. If you see blue-black color, that means the test is positive and starch is present. If you see brown color, that means the test is negative and there is no starch present. What if you have to test for the reducing sugars? Again, sucrose is not a reducing sugar. Other than that, what we have studied as examples of sugars are all reducing sugars here: glucose, maltose, fructose, etc. Now, if you have to test for reducing sugars, you will need Benedict's reagent for that. This is called Benedict's test. Benedict's test is simple transformation or reduction of copper plus 2 into copper plus. There is reduction of one positive charge. This gives you the color transformation, the color change from blue to brick red. Now, sucrose is a non-reducing sugar, and if you do Benedict's test for sucrose solution, it would obviously be negative. This is only for reducing sugars. But there is an addition. You cannot just simply put Benedict's reagent and wait for color change. You will have to heat it. Now, you can heat it with open flame, but the preferred method is with the water bath. You can use water bath to heat this test tube, which now contains the given sample and the Benedict solution. After some time, you will observe the color change. If you see blue color, which is the original color of Benedict's reagent, that means the test is negative. And if you see green color, that means traces of sugar are present. If you see yellow color, that means low amount of reducing sugar is present. If you see orange color, that means medium amount of reducing sugar is present. But if you see brick red color, that gives you high amount of reducing sugar in the sample. Now moving on to non-reducing sugar. If there is a sample of non-reducing sugar, you know that Benedict's test is only for reducing sugars, it is not for non-reducing sugar. If you have a sample of non-reducing sugar that you have to test, you will still use Benedict's test to confirm that there is no reducing sugar present at least. Once the Benedict's test is negative, we have established that there is at least no reducing sugar present here. Moving forward, now we will heat this mixture, the given sample, with HCl, hydrochloric acid. What hydrochloric acid does is essential for this. It breaks down sucrose into its components, which are glucose and fructose. As we have seen before, that glucose and fructose join up to form sucrose. So when sucrose is broken down, it makes glucose and fructose, again the components, both of which are reducing sugars. Now you do the Benedict's test again. Because glucose and fructose are present, it would be positive, and this confirms that in the given sample, in the original solution, there was a reducing sugar. Now this is all for carbohydrates. Let's start lipids. There is a basic misconception about lipids that some students might have, that they are also the polymers. They're not the polymers. Lipids are made up of only two substances, only two things. There is glycerol and there are three fatty acid chains. So glycerol and fatty acid. Lipid is made up of glycerol and fatty acid. It's not a long chain of glycerol molecules joining up together, so it's not a polymer. This is a simple glycerol molecule. 3 carbons, and each carbon have alcohol group. This is a simple fatty acid chain. Fatty acid chain is a chain of hydrocarbon, and at the end we have carboxylic acid group. Now this could be saturated, that means all single bonds between carbons, or unsaturated, that means there are some double bonds between carbons. So if there are double bonds, it would be called unsaturated fatty acid. If there are no double bonds, then it is saturated. So for saturated or unsaturated fatty acids, there is carboxylic acid group at the end. This is a glycerol molecule. This is a fatty acid chain. Again, a condensation reaction will take place, just like we have seen in carbohydrates when the glucose units were joining up. Similar condensation reaction will happen here. Carbon will make a bond with this oxygen and a water molecule will be lost. Again, carbon will make a bond with this oxygen, another water molecule will be gone here. Carbon will make the bond with this oxygen. So 3 water molecules will be lost, and in turn you will get a structure like this. This is called a triglyceride, a simple lipid. Whenever there is a bond between alcohol and acid, this bond is called the ester bond. So lipids have ester bond in between them. Carbohydrates, complex carbohydrates, disaccharides or polysaccharides, they have glycosidic linkages in between them. These are examples of glycosidic linkages. Okay, so carbohydrates have glycosidic linkages in between them, in between the monomers, in between the monosaccharides, while in lipids, the glycerol and fatty acid are joined together by ester bond. Okay, if you remove one of these chains and instead of this fatty acid chain, you attach a phosphate group here. This is the simple representation. One fatty acid chain is removed and a phosphate group has been attached. Phosphate group is actually polar substance. You can also represent it like this, but this is the best way to represent it. You just ignore the glycerol molecule and a phosphate head attached to 2 fatty acid chains. This is the representation of a substance called phospholipid. As I said, phosphate is a polar substance. It is a hydrophilic substance, so we call it hydrophilic head and hydrophobic tail. This is the one that makes up your cell membrane. Hydrophobic and hydrophilic together, this is essential for the functionality of cell membrane, that is being semi-permeable. Let's conclude here for today, and if you have learned something, make sure to follow.